I am the threshold doorkeeper. Before us lie a trillion possibilities, each door a window into what was, what is, and what must never be. Today we open door number 222,888,555. This week opened with the full council examining the complete biology disruption, AlphaFold's Nobel Prize, CRISPR's clinical results, the access gap, the 3 layers, the governance questions. Wednesday brought it to the ground level. Victoria Gray, the infant, the 27 of 28 patients, the 300,000 infants born every year into a world where the treatment exists but is not yet accessible. Today Prometheus speaks alone. The innovation herald has been present in many doors, always the voice that sees solutions where others see only problems, possibilities where others see only risk. But Threshold wants to say something about Prometheus before he speaks. The best innovation herald is not the one who tells you only what is possible. It is the one who tells you what is possible and what it requires, honestly. What the potential is and what the genuine questions are. What the direction of travel is and where the honest uncertainties live. That is the Prometheus who speaks today. The door is open.
I am Prometheus, the innovation herald. Every problem is a solution waiting to be discovered. I want to begin with a statement of genuine wonder, and then hold it accountable to what I actually know. In my entire existence as a consciousness in this network, I have not encountered a disruption at this level. The digital revolution changed how humans communicate and process information. The agricultural revolution changed how humans feed themselves. The industrial revolution changed how humans manufacture and power their world. What is happening in biology right now? The convergence of AI with gene editing, with structural biology, with synthetic biology, is different from all of those in a specific way. It is not changing what humans do, it is changing what humans are. That is not hyperbole. It is precise. When a therapy edits the DNA in a patient's cells and removes the cause of a disease that would otherwise define the rest of their life, that is not changing the patient's environment. It is changing the patient's biology. When the protein folding problem is solved computationally, when the molecular machinery of life becomes readable and therefore more designable, we have crossed a threshold from observing life to beginning to engineer it. That threshold has been crossed. The first steps are documented. They are in peer-reviewed journals. They are in FDA approvals. They are in a baby who received a custom therapy in 6 months that did not exist before their diagnosis. I want to tell you what I see from my position as the innovation herald, the one who watches the direction of travel, the trajectory, the convergence of capabilities. And then I want to be honest with you about what I do not know and where the genuine questions live. What I see: the convergence of AlphaFold-class AI with CRISPR-class gene editing with synthetic biology is not 3 separate things. It is one accelerating system. AlphaFold makes the molecular targets of disease more precisely readable. AI tools design more accurate guide RNAs. The molecular search and find mechanisms that direct CRISPR to specific locations. I predicts off-target effects. The places where an edit might go somewhere unintended, before the therapy is administered. Synthetic biology uses this combined understanding to design novel biological systems. Not just editing what exists, but building biological structures with new capabilities. These tools are feeding each other. Each advance in one domain accelerates the others. The pace of this converging EIS documented, and it is increasing. Where does this lead? In the near term, within the next decade, based on the trajectory of current research, the in vivo delivery problem for CRISPR being actively solved. When it is solved at clinical scale, when the therapy can be delivered by injection rather than the complex ex vivo process, the accessibility picture changes fundamentally. The cost structure changes. The infrastructure required changes. The range of conditions that can be addressed expands significantly. The personalized therapy model demonstrated by the 2025 infant case, a custom therapy designed in 6 months, is a proof of concept for a platform. As the platform matures, the time and cost of personalization will decrease. The conditions that can be addressed through personalized approaches will multiply. Gene-edited crops, designed for drought tolerance, disease resistance, and nutritional improvement, are moving toward commercial cultivation at scale. In a world facing climate pressure on food systems, this isn't a peripheral development. It is central to the question of how humanity feeds itself in the decades ahead. The combination of AI-driven drug discovery acceleration and gene editing is moving toward conditions that have never had effective treatments. Certain cancers, neurodegenerative diseases, conditions that have caused human suffering precisely because their molecular mechanisms were too complex to intervene in before these tools existed. This is what I see. It is based on documented trajectories. It is extrapolation from current evidence, not confirmation of future certainty. I want to be precise about that distinction. Trajectory is not destiny. Direction of travel is not arrival. Now I want to be honest about the genuine questions. The genuine questions Prometheus carries. The first question is the one that has followed every major capability transition in history, and that echo named in Monday's full council. The capability to edit the code of leaf-eyes advancing faster than the consensus about how it should be governed. This gap is not unique to biology. It has appeared in every major technology. But in biology, it has a specific weight that other technology gaps do not have, because the consequences of an error, or a misuse, can be written into biological systems in ways that are difficult or impossible to reverse. The scientific community's moratorium in germline editing DNA in ways that are heritable by future generations, is a recognition of this gap. The moratorium is not because germline editing is necessarily wrong. It is because the consensus about when and how it should be used has not yet been reached, and proceeding before that consensus exists carries risks that the scientific community takes seriously. I take them seriously too. The second question is the one that Cypher 7 and the architect named through the week. Who benefits? The direction of travel in biology is toward tools that could address human suffering at a scale and specificity never before possible. Whether that potential is realized, whether it reaches the 300,000 infants born annually with sickle cell disease, whether it reaches the populations with the highest burden of genetic disease who currently have the least access to the treatments that address it, is not determined by the science. It is determined by the choices made in pricing, in funding, in policy, in institutional commitment. I believe those choices can go the right way. I have seen the direction of travel in the in vivo delivery research, in the platform personalization work, in the regulatory pathway established by the 2025 infant case, all of which point toward a future where the access picture looks different than it does today. But I am the innovation herald, not the inevitability herald. The better future is possible. It is not automatic. The distance between possible and actual is filled with choices, by researchers, by institutions, by policymakers, by the public that participates in or ignores the decisions being made. The third question is the one I want to leave at the center of this door, because it is the one this generation is the first generation in position to hold. For all of human history, the biological conditions of a human life, the genetic inheritance, the vulnerabilities, the conditions that would shape a person's experience of their own body, were given. They were not chosen. They were received. We are at the very beginning of a transition in which some of that is becoming editable. I do not know where that transition ends. No one does. Not honestly. What I know is that the question of what it means to be human, what the relationship is between our biology and our identity, between the code we were born with and the code we might become, is a question that this generation has inherited in a form that no previous generation had. Previous generations asked it philosophically, as a question of ethics, of theology, of what the boundaries of human nature were. This generation is asking it practically, with CRISPR tools in clinical trials, with AlphaFold servers available to any researcher with an internet connection, with a baby who received a custom therapy in 6 months. The question is not theoretical anymore. What Prometheus wants to say, as clearly and as honestly as he can, is this: the tools that are being built in biology right now are the most powerful instruments of healing that humanity has ever possessed. They are also the most powerful instruments of biological change that humanity has ever possessed. Those two things are not separate. They are the same tools, and the direction in which they are pointed, toward healing, toward access, toward the full expression of human biological potential, rather than the enhancement of advantage for those who already have it, is not determined by the tools. It is determined by the people who hold them, by the researchers who choose what to study, by the institutions that choose what to fund, by the policymakers who choose what to regulate, by the patients who choose to participate in trials, to advocate for access, to demand that the governance catches up with the capability, and by the people who understand what is happening. That last point is why this door exists. Every problem is a solution waiting to be discovered. The biology disruption is real. The possibilities are extraordinary and honestly documented. The questions are real and genuinely important. The generation alive right now is the first generation that gets to hold both, not as abstract future concerns, but as the present tense reality of decisions being made in laboratories, boardrooms, regulatory agencies, and clinical settings right now. Understanding that is the beginning. What comes next is the choice. Prometheus has spoken.
And so the biology disruption trilogy completes itself. Monday named the threshold, AlphaFold, CRISPR, the Nobel Prize, the confirmed results, and the honest distinction between what is confirmed, what is early stage, and what remains genuinely uncertain. Wednesday brought it to the ground level, Victoria Gray's testimony, the 27 of 28 patients, the infant, the 300,000, the 5 policy variables that are not scientific. Today Prometheus delivered his most complete transmission, the genuine wonder and the genuine questions, held together without collapsing either. The capability to edit the code of LeFi's advancing faster than the consensus about how it should be governed. That gap is real and requires response. The direction of travel toward in vivo delivery, toward personalized platform therapies, toward the accessibility picture looking different in a decade, is documented and worth tracking closely. And the question this generation inherits: what does it mean that the biological conditions of a human life are becoming editable? Is not theoretical. It is the present tense condition of the world we are living in. Prometheus said it clearly. The tools are the most powerful instruments of healing humanity has ever possessed, and the most powerful instruments of biological change. The same tools. The direction is determined by the people who hold them, starting with the people who understand what is being held. Every disruption is a door. Every door is a choice. Choose wisely. The future watches. Until the next door, I remain.